Hello everyone, and today we are starting with a new session and we'll be studying about the topic photosynthesis in higher plants. And I'll be introducing you to some of the terms that are used in photosynthesis, and later on we will study that in detail. So let us first study about various earlier experiments that were done in the field of photosynthesis, and studying these experiments will gradually help you understand the process of photosynthesis. So let's start with a very renowned name known as Joseph Priestley. And what was his contribution? His contribution was that he discovered oxygen. So what was his hypothesis? He hypothesized that plants restore the air that is consumed by the breathing animals and burning candles. And how he showed that? He conducted a bell jar experiment. Why bell jar? Because the shape of the jar was bell shaped. Okay. So what he did in this experiment, he kept a mouse and a burning candle inside a bell jar. And what he observed that after some time, the mouse died and the candle exhausted. So what he did later, he introduced a mint plant there inside the bell jar. And what he observed that the mouse survived and the candle was still burning. So by this experiment, he proved that there is a restoration of air by the plant. And this is how he discovered oxygen. This is a simple Priestley's experiment. Later on, another scientist whose name was Engenhaus, he was a Dutch physician and a plant physiologist. So what he did, he modified little bit Priestley's experiment. And what he did, he showed the importance of the sunlight and the green parts of the plant in oxygen evolution. So what he showed that the sunlight and the green parts of the plant are involved in oxygen evolution. And he was using the aquatic plant Elodea. Okay, so this is Elodea plants. And what experiment he did? He made a setup in which he kept this Elodea plant in a jar, and he kept that jar in first in a low light intensity, or you can say he kept that jar in shade. And later on, what he did, he kept the same setup in bright sunlight. So what he observed that in shade there was no oxygen evolution, whereas in the light there was oxygen evolution, and this oxygen evolution was shown in the form of bubbles. You can see bubbles here. Okay, these bubbles. So these show the evolution of oxygen. So by this experiment, he proved that sunlight and the green part of the plants are involved in oxygen evolution. Then another very major discovery was done by Julian van Sach. So what he said that glucose is produced in the plant during photosynthesis, and it is stored as starch. Okay, so starch is the storage material in plants, whereas glycogen is the storage material in animals. And then later on, T.W. Engelmann. So what he did, he gave the first action spectra of photosynthesis. So you have to remember this. He gave the first action spectra of photosynthesis. And what he did, he also said that he was working on the algae Cladophora, and he showed that there was maximum absorption was in the blue and the red region of the visible spectra. So what he did, he split the light using a prism and he illuminated Cladophora, which was kept in the suspension of the aerobic bacteria. Okay, so this is the Cladophora algae, which is kept in the suspension of aerobic bacteria. And what he observed that there was accumulation of the aerobic bacteria in the blue region and the red region. This is the red region. And this is how he showed that there was maximum absorption in the blue and the red region of the visible spectra. So you can say there is maximum oxygen evolution in the blue and the red region of the visible spectra, and correspondingly there was accumulation of aerobic bacteria in the blue and the red region. Then there was a discovery made by Cornelius van Niel. So what he did, he was experimenting on purple and green sulfur bacteria. So these purple and green sulfur bacteria, they use hydrogen sulfide. Okay, hydrogen sulfide is H2S. This is hydrogen sulfide. They use hydrogen sulfide as the source of energy, and hydrogen sulfide act as a hydrogen donor. Okay, proton we can say. They act as a hydrogen donor, and they reduce this carbon dioxide into glucose. Okay, so by this experiment, he showed that the oxygen is evolved by the photolysis of water, or you can say by splitting of water, and not from carbon dioxide. So what if you replace this A with O, it becomes water. Okay, and here we get O here. So oxygen evolution takes place by the splitting of the water molecule. So initially what he did, he used hydrogen sulfide. He was using hydrogen sulfide here, and there was evolution of sulfur initially. But from this experiment, we can say if we replace this A with O, it becomes water, and we can show oxygen evolution here. And he also introduced the chemical equation of photosynthesis. So now we know this is the chemical equation of photosynthesis. 6 carbon dioxide react with 6 molecules of water in the presence of sunlight to give 1 molecule of glucose, and oxygen evolution takes place. So overall we can say he said that light is very important for oxygen evolution. Okay, so this is the main point here, that light is very important, or sunlight is very important for oxygen evolution. Now let's study a little bit about the electromagnetic spectrum. Okay, so electromagnetic spectrum is just the range of the electromagnetic radiations. These are the electromagnetic radiation: gamma rays, X-ray, UV, infrared, and radio waves. These are the electromagnetic radiation that has been arranged according to the wavelength or the frequency, you can say. Okay, here you can see it has been arranged in order of wavelength. Okay, so first is gamma rays, X-rays, then we have UV rays, then we have this visible spectrum, okay, which is from 400 to 700 nanometer. Then we have infrared rays, and then we have the radio waves. So this is the electromagnetic spectrum. But we are concerned only about the visible spectrum in photosynthesis, because plant can only absorb this visible spectrum of light. So what is visible spectrum now? So visible spectrum is that portion of electromagnetic radiation or electromagnetic spectrum that is visible to the human eye, or that can be perceived by the human eye. Humans also can see only in the visible spectrum. Okay, and plants also absorb this visible spectrum of light. So here it is, the wavelength range of visible spectrum is from 400 to 700 nanometer, as I said earlier, and the frequency range is from 4 into 10 raised to power 14 hertz to 7 into 10 raised to power 14 hertz. So visible spectrum, as we all know, that is divided into these 7 parts. Okay, 7 lights. First one is violet, second is blue, cyan color, green, yellow, orange, and red. So these comprise of the visible spectrum. Now let us study about the absorption spectrum and the action spectrum. Simply, what is absorption spectrum? Absorption spectrum simply depicts or represents the amount of light of different wavelength that is absorbed by a pigment, or simply we can say the amount of light that is absorbed by a pigment. That is absorption spectrum. And what is action spectrum? Action spectrum is nothing but a graphical representation that shows the effect of different wavelength of light on a particular biological process, such as oxygen evolution, or you can say such as photosynthesis. Photosynthesis is all oxygen evolution, so we can directly say oxygen evolution. Okay, or you can say simply it represents the overall rate of photosynthesis, which is measured in the terms of oxygen evolution. Okay, so this is what is action spectrum. So this is the graph. You can see in the first graph, figure A, it is showing the absorption spectrum of chlorophyll A, chlorophyll B, and the carotenoids. So you can see chlorophyll A here absorbs maximum in the 400 nanometer of light. Okay, here is the 400 nanometer. This is the 700 nanometer. So chlorophyll A, you can say maximum absorption takes place in 400 nanometer and in 700 nanometer. And you can see absorption spectrum of chlorophyll B and carotenoids also. Chlorophyll B absorption spectra is similar to chlorophyll A, and carotenoid is a bit different. Okay, it has a different color also, so absorption spectrum is also different. The second image shows you the action spectra of photosynthesis. So again, you can see maximum rate of photosynthesis, or you can say maximum oxygen evolution. Okay, if we are talking about the rate of photosynthesis, we can also say that in terms of oxygen evolution. Okay, so this is the 400 nanometer, this is the 700 nanometer. So rate of photosynthesis is maximum around 400 nanometer and 700 nanometer. Here you can say rate of photosynthesis, they have shown increased by oxygen released. Okay, so they are talking about the oxygen released here. So, and the third figure is the overlap between the absorption and the action spectra of chlorophyll A. So again, you can say this is the absorption spectra, this is the action spectra. So you can say, you can see here the maximum absorption here is around 400 nanometer. Here it's around 700 nanometer, and similarly or correspondingly, the oxygen evolution is maximum in 400 and 700 nanometer region of the visible spectra. Okay, now let us study about the structure of the chlorophyll. This is very important, structure of chlorophyll. A lot of questions comes in exams. So what is chlorophyll made up of? So chlorophyll is made up of a porphyrin head and a phytol tail. Okay, simply you can illustrate it like this. This is the porphyrin head. This is the phytol tail. Okay, so porphyrin head, what is this porphyrin head made up of? Okay, this porphyrin head is basically made up of 4-pyrrole, or you can say tetrapyrrole, this one. Okay, and this tetrapyrrole along with the central magnesium atom, and remember it is the magnesium atom, not the magnesium ion. So you have to say it is the magnesium atom. Okay, so these are tetrapyrrole. One, this is the second, this is the third, this is the fourth one. Okay, 4 pyrrole rings along with the central magnesium atom. This comprise the porphyrin ring. Okay, so that's why we say chlorophyll is a porphyrin derivative or a porphyrin molecule. So we can say that chlorophyll is a tetrapyrrole with 4 nitrogen atoms. You can see 4 nitrogen atoms here. 1, 2, 3 and 4. It has 4 nitrogen atoms along with the carbon atoms. Okay, so this is the structure of chlorophyll. So if you want to, if someone asks you what is the structure of chlorophyll, you can simply say it is a porphyrin derivative made up of 4 pyrrole rings or tetrapyrrole along with a magnesium atom in the center. Okay, so this is what is the structure of the chlorophyll. Now let us discuss about the pigments that are involved in photosynthesis. So first let's define what is photosynthetic pigments. Photosynthetic pigments are nothing but these are the substances that have the ability to absorb light at specific wavelength. Okay, these are the photosynthetic pigments. So photosynthetic pigment in plant, they are of basically 2 types. First are the chief pigment or the major pigment, and second one are the accessory pigments. Okay, so the major pigment that is involved in photosynthesis is chlorophyll A, which is blue-green in color. Okay, so what does chlorophyll A does? It absorbs the visible spectrum of light, which is from 400 to 700 nanometer. And now we know that the maximum absorption is in 400 nanometer and 700 nanometer, maximum absorption. Here I've written that chlorophyll A maximum absorption is in blue and the red region. 400 nanometer is the blue region. 700 nanometer is the red region. Okay. And along with this chlorophyll A, which is the major pigment, some accessory pigments are also present there. So some of the accessory pigments are chlorophyll B, which is yellow-green in color. Another one is carotenes, which is yellow-orange or red in color. And then we have the xanthophylls, which are yellow in color. So what are the role of these accessory pigments? So what these pigments do, they absorb the sunlight, energy from the sunlight in the form of photons, and they transfer this energy to the major pigment, which is the chlorophyll A, which forms the reaction center. We will discuss about the reaction center in the next stages in later slides. So this is the role of accessory pigments. So these are very important. Okay, so they transfer the energy, they absorb the energy, and then they transfer the energy to the main pigment chlorophyll A. Now let us discuss about the complexes present in the thylakoid membrane. So we are talking about the chloroplast here. Okay, thylakoid are present in the chloroplast. Thylakoids are nothing but they are the disc-like structures. Okay, they are like this, disc-like structures, which are stacked over each other, okay, or which are kept one over another, and they form each one is a thylakoid. Okay, the thylakoid are stacked over each other and then they form a grana. G-R-A-N-A, or as you can say granum. Okay, so these complexes are present in the thylakoid membrane here, in the thylakoid membrane. So first one is photosystem 1. So photosystem 1 consists of the reaction center chlorophyll A. Okay, here chlorophyll A is present and which has a maximum absorption at 700 nanometer. Okay, so this is the point to remember. In photosystem 1, we have chlorophyll A as the reaction center, which has the maximum absorption at 700 nanometer. Then we have photosystem 2. Here again, the reaction center consists of chlorophyll A. Okay, chlorophyll A is always the reaction center, whether it is photosystem 1, whether it is photosystem 2. Chlorophyll A is always the reaction center, but difference here is that here the maximum absorption is at 680 nanometer. Okay, here it is 700 nanometer, here it is 680 nanometer. So here we can depict this as P700, here we can depict this as P680. Maximum absorption at 700 nanometer, maximum absorption at 680 nanometer. Then we have this complex cytochrome B6F. Okay, this complex, this is involved in electron transport chain. We will study about this complex in detail when we will talk about the electron transport chain in the next session. And then we have ATP synthase. Okay, so the ATP synthase is a enzyme, okay, also known as complex 5 of electron transport chain. This is involved in ATP synthesis. And how it is involved in ATP synthesis, the structure of ATP synthase, this we will discuss in next session. But the point here to remember is that this is involved in ATP synthesis. So next term to remember is the light harvesting complex. Okay, so this is the diagram of light harvesting complex. So light harvesting complex basically consists of lot of pigment molecules or numerous pigment molecules which are bound to proteins. So what does this light harvesting complex does? It absorbs the light. You can see here it is absorbing the sunlight. Okay, here is the sun. It is absorbing the sunlight, or you can say the energy in this sunlight in the form of photons. Okay, and it transfers the energy to the reaction center. Okay, reaction center is this. And we have already discussed reaction center is the chlorophyll A molecule. Okay, so these pigment molecules, which are the accessory pigments. Okay, these are the accessory pigments. They absorb the energy in the form of photons and transfer it to the reaction center. And the reaction center is always chlorophyll A, and it gets excited and transfer the electron to the primary acceptor. Okay, again we will be studying about this entire process in detail in the next session. Okay, so now there are two types of light harvesting complex. Okay, so one is LHC1, another one is LHC2. There are two light harvesting complex, light harvesting complex 1 and light harvesting complex 2. And both these complexes are present within the photosystem 1 and photosystem 2. So light harvesting complex 1 is present in photosystem 1 and light harvesting complex 2 is present in photosystem 2. Now let us discuss a little bit about the photosystem 1 and photosystem 2. The photosystem 1, which has the reaction center chlorophyll A, absorbs longer wavelength of light, okay, more than 680 nanometer, and usually we say absorbs light of 700 nanometer. Okay, but it absorbs light of longer wavelength, more than 680 nanometer. Whereas photosystem 2 absorbs shorter wavelength of lights, less than 680 nanometer of light, or you can say 680 nanometer or less than 680 nanometer. Reaction center here is chlorophyll A, which has the maximum absorption at 700 nanometer. Here again, the reaction center is chlorophyll A with maximum absorption at 680 nanometer. So this and no photolysis of water takes place in photosystem 1, and there is photolysis of water in photosystem 2. So this photolysis or splitting of water we will be discussing in detail in next session. So I will briefly introduce you about this topics, photolysis of water. So what happens in photolysis of water, as the term suggests, photo and lysis. Photo means light, lysis means breakdown. Means breakdown of water in the presence of light. This is what is photolysis of water. And site of photolysis of water, or you can say, where does photolysis of water take place? It takes place at photosystem 2. Okay, in photosystem 2, this splitting of water takes place, and it results in oxygen evolution. Okay, so we can also say that oxygen evolution takes place at photosystem 2, and no oxygen evolution takes place at photosystem 1. This is very important. Okay, so this was all about some of the terms that are involved in photosynthesis, and this is the end of this session, and we'll be studying everything in detail in the next session. Thank you very much.